Okay, so got different slides up on the computer. So the main thing here is the final word properly. That I want to do these things, do these analyses properly, and I'm going to argue, perhaps incorrectly, that AI is not the answer. Sorry, folks. And first, I want to acknowledge that my job here is to be the pretty face, so already a failure. These guys did all the hard work. I'm just representing it. I'm just trying to take the credit for it. And they have done a lot of hard work here. It's been great. The problem is that biodiversity data is messy. Some people don't see this because we sort of try to hide it, or people ignore the messiness. But it's messy, and if you get over the messiness, there's a lot of useful information in there. And so, for example, GBIF here is a public database. The data is all open. It has 3 million observations of 1.4 million species. So I think we're pretty much in the big data area here. And our challenge is to use this data to map where the species are, and then obviously from that, using a model of where they are to predict where they'll be in the future. And there's all sorts of reasons for doing this. I'll explain those later. Just to give you an example of the problems, this is a map of the observations of mammals in Norway, and this is not humans. Humans are excluded from this. The darker the color, the more mammals there are, and somebody's already seen the punchline. So there's a blob down here, there's a blob up here. I don't know if you've noticed, there's also a line up here. And for those of you who don't know Norway, the blob at the bottom is Oslo, capital city where Marjan lives. This blob in the middle is Trondheim, which is where I live and where there's a lot of environmental research as well, both from the university, and there's a few institutes around there. And this line down between them is the E6 motorway. And I'm going to suggest that this is not the distribution of mammals in Norway. And I think all of the hunters in Norway will agree with me. So this is the problem, that most of the data, a lot of what we see, if we just map them like this, is due to human activity, not due to where the species actually are. So that gives us a problem. If you want to model where the species are, we have to separate out the observation process from all of the ecological processes. What makes it either better or worse is that data sets are different. You've heard earlier talks about a lot of the monitoring work and using things like camera traps. Really good talk about them. The other forms of monitoring, so there's mention of the Chinese equivalent of the Christmas bird counts, where you produce checklists of the birds that you see in your garden. And there's more structured forms of sort of ground-based observations like that. The breeding bird surveys, butterfly people do things similar. There's all sorts of things checking like that. Somebody mentioned malaise traps. So if you're catching insects, you put out traps in one way or another, which are all slightly different. There's also modern techniques like eDNA, which are really going to change the field and make things even messier. And then there's citizen science data, where you just get observations. So you take a photograph of a species that you've seen, you upload it to iNaturalist, it gets identified, and then we'll go through to GBIF. This is all great data. It's all sort of useful, but it's useful in different ways. So these are great, but how do you expand out? Well, I'm going to suggest that there's so much other data around that will help you expand out. Use the monitoring data, it's high quality, to tell you what's going on in some areas. And you can use difference between that and, say, citizen science data to look at the biases in the citizen science data. And we formalized that, which I won't go into. So given those problems, we've been building our solutions to be able to model these distributions using sort of this big, messy data. And now I will wind these guys up at the front here by saying AI is not the answer. They can show me wrong. It'd be really interesting if they did. There's a few reasons. One is that what we're interested in is internal to the model. It's not the data itself, because I've told you that's a horrible mess. At the other end, we're going to use predictors, things like climate variables, things from remote sensing satellites to try and predict the distributions. Once we've removed the observation biases, then there's something internal, the actual distribution, and that's what we're going to be interested in. I'm not sure AI can extract that out, but it'd be fascinating to find that out if I'm wrong. The other reason I don't think AI is the answer, which links to the previous talk, is that we have a lot of ecological knowledge. We've been modeling the species distributions, the species dynamics for a long time, often using data. We know have a good idea about how the species distributions change. The problems are the details and scaling it up. We also know other problems like spatial autocorrelation, which can come out partly through things like dispersal, but also because we know we're not measuring the right variables. There's always going to be some residual correlations which are going to have an effect. So all in all, my argument is that we know enough to know that there are these things that are cropping up, going to crop up in the model that we need to deal with. And AI hasn't yet learned that we need to know about these, whereas we have, so we can put them into our models. The structure of our solution we're taking looks something like this. It's a hierarchical structure. So in the middle, we have the actual species distribution. That's what we're interested in. We're assuming ecologically it's being driven by things like climate change, land use, any that we might have, for example, interactions with other species, other variables, perhaps the soil type, that sort of thing. So that's the ecology at the top. That's what we're really interested in. At the bottom, we have the data. Okay, I'd say that's what we're really interested in. I'm a statistician. The data is what I'm really interested in. Different data sets. I said there are different types, they have different strengths and weaknesses. So me as a statistician is sat there going, well, let's try and put these together so that we can get the strengths from all the different data sets, data types. So the data sets are generated by the actual species distribution and all the various human biases we put into it by, for example, only observing mammals when you're driving up the E6. So that's the structure. How we do this is we think about our species, individuals being points in space. We then think, okay, they're drawn from some distribution, which we model to continue in continuous space. And we use something called an inhomogeneous Poisson point process. We do this because there's lots of really nice maths which we can use. So, for example, in any area, the number of points follows a Poisson distribution. And we can just manipulate that mathematics. So here's some of that really nice helpful mathematics. I will not go through this slide. I'll let you take a photo. There's more. This is only part of it, of course. And if I can get on to the next slide. Yes. What are we doing with all this maths I've not told you about? So we've developed the models. We've been able to, because it's not that difficult to write them down. Then coding up in software, we're trying to code them in a way where it's easy to use. So you don't have to do all the complicated coding yourself. There's lots of details with this, particularly getting working efficiently. So Philip, the first author on this paper, spent a lot of time working out how to do this, how to split up the analyses in different jobs. Again, this gets back to big computing, how to do that well. We've also developed pipelines. So this is going to work linking with BioClima to get the data, push it through this pipeline, and get a variety of products, which might just be maps of where species are, or perhaps maps of species richness. But we can develop all sorts of other products. Our products could also be projections into the future, so predictions of what's going to happen under climate and land use change. Again, a ton of work done to do this. Now I'm going to talk about one specific project that we were asked to work on. It's called the Hotspots Project. So we were asked by the Norwegian Environmental Agency to look at hotspots of biodiversity in Norway. Now, once we'd stopped laughing at the idea that Norway has hotspots of biodiversity, we realised they want it relative to other bits of Norway. So they asked us to look at several species groups, which was over 8,000 species in all, going from, say, 9 bat species up to over 4,000 insects. This also included birds, different mammals, vascular plants, etc., fungi, lichens. And for each one of these, we selected about 10 covariates. So we didn't do this by AI. We did this by asking the experts in the field who knew about the species groups. We were told we had to use open data, which was good because we wanted to anyway. So we're using GBIF, which I mentioned before. And really annoyingly, they said we had to do this at the 500-meter scale, which is a really fine scale, getting down to the point where it's not really ecologically meaningful. That just means that in practice that lots more computation time. So we scaled up this all up, said a lot of hard work to do this, to be able to push through 8,000 species through this without having to look at each one individually. After a lot of hard computing, the good news is that the computer we did this on is located in Trondheim and it's used heat of NTNU. So the reason why it's so hot in NTNU is because of what we were doing. And we get products like this of species richness. So we can see here in the southeast, the green means a high species richness, much lower to the west. That's because that's the high mountains and glaciers. So we expect to see that. So this looks reasonable. We can also get the distribution's covariate effect. So for example, in birds, we can read this off and see that there's a distribution of effects of summer temperature, but on average, it's positive. So what next? I'm suggesting world domination. We did this for Norway, but people here have been talking about doing similar things. I've been talking to people in Australia, in Canada, in South Africa, who are also interested in doing this. And what I'm suggesting is that we should have one pipeline to do this. And we should standardize it so that we can push to data from Norway or from South Africa or Canada or Australia through these pipelines together. If we do this, it means that we only have to develop them once. And there's some people interested in this. We can get the experts all around the world to find the bits that they're interested in and really develop them. So let's do this together. And as a part of this is the reason why I'm in BioClima, because we have some really great Europeans here who can help us with this, as well as, of course, many of you from China. So, thank you.